Hello all, welcome back to the BioRocks channel. As you can see it on the screen, and it is evident that the topic today that I'm going to deal in this video is going to be about the fertilization. But before I define, I enter into the video, children, I would like to make it clear that this is one of the long processes, the fertilization process. So I'm breaking this fertilization process into the series of two different videos, the part one and the part two, to make it easier for you children to understand it far better. So without any delay, I'm starting the process of fertilization. So as you can see it on the screen, it is just like a fertilization can be defined as a fusion of the male as well as a female gamete. As you can see, the male gamete is going to be the spermatozoa or the sperm cell, as you can see, and the ovum, the haploid female gamete, togetherly is being called as a fertilization. And this fertilization happens in the female reproductive tract. It is an internal fertilization that happens in the ampulla area of the fallopian tubule. Okay, children. So this continues, or this is going to be a very, very long process. So what I'll do is first let me take up the female reproductive tract and show its journey. Then I'll enter into this topic detailedly, going through the point wise. So this is going to be the vagina, the long muscular tube, the birth canal, where the ejaculation happens of the semen along with the sperms. If you look at 1 ml of the semen, it is going to be in 1 ml of semen, it's going to be the only 10% of it is going to be the sperms, and the 90% is going to be the fluid composition with all the other components like prostaglandins, the fructoses, and the different several enzymes which are being present. Okay, during an ejaculation, a male can ejaculate from 2 ml to that of 5 ml, and each ml of the semen comprises of some 100 ml, 100 millions of sperm. So together it might be 300 million sperms if it's going to be 3 ml of the semen being ejaculated into the vagina after the or during the intercourse or the coitus. If it has been ejaculated, you can see the abundance of sperms being ejaculated into the vaginal area, where they have to move through the cervical opening into the long uterus tube, that is muscular tube called as uterus. And then you can see that on either side of your uterus we have the fallopian tubules. Okay, we can even call them as uterine tubules, and these are going to have the bridge-like connections called as isthmus. They have a wide and a very long tubes called as ampulla area. And you can even see that the conical shape here is going to be the infundibulum. And these are the fimbriae which are being present. This is one of the beautiful images where you can see that the fimbriae are very near on the surface of these ovaries. I will speak more detailedly about this, and before I end the video also, once again I'll come to this topic of fimbriae being brushing up the surface of the ovary. Okay, so these are the parts. If you see, the ova gets released from the Graafian follicle during the ovulation process, which is actually going to be a secondary oocyte with a few of the layers of the granulosa cells, the follicle cells, and with little amount of fluid, it is going to pop out, received by these particular fallopian tubules here, children. And they have to travel only towards the ampulla area where they get fertilized. Okay, this is the ampulla area here, what they're showing. So this is going to be the ampulla area. Okay, but if you look at the journey of the sperms, it's very long. They have to travel from the vagina through the cervix into the uterus, then move into the fallopian tubules and reach the ampulla area for fertilizing the female gamete. Okay, children. So let me take this procedure into a stepwise manner such that it will be easy for you all children to understand. So for that, what I'll do is I'll take up my whiteboard to explain you all these procedures step by step. As I earlier told that I'll be making such interesting videos for the NEET and for the boards. And especially today, the video that I'm making is going to show shortly in ensure that you will not miss a single point in your medical entrance examinations, because I am going in a very detailed manner, due to which I have broken the fertilization video into two parts, and this is going to be the first part. First thing, speaking about what is being called as fertilization process here. What is fertilization? I told you this can be defined as a fusion of male and female gametes. Fusion of male and female gametes. Male and female gametes. So what are those male gametes? It's going to be the sperm, and this is going to be your ovum, or what we call as egg. Okay, so this egg is actually in a stage of a secondary oocyte. Actually, it's a secondary oocyte being enclosed by a glycoprotein layer, which is being called as a zona pellucida. This is going to be the zona pellucida, and you are going to have another, what you call is, it's being surrounded by a few of the follicular cells like this, and these follicular cells are going to be called as what? It's going to be called as the corona radiata. Corona radiata. And we will see the procedure. I'll even speak about this also. But just remember that it is a fusion of this sperm as well as the ovum or the egg, which is being called as fertilization. Then we will speak about, in humans, it's going to be internal fertilization, right? So in the humans, the type of fertilization is going to be called as the internal fertilization. Internal fertilization, since this is happening inside the female reproductive tract, that too in the area of the fallopian tubules called as ampulla. So this is an internal fertilization. Then next, coming on to the person to describe the fertilization in the sea urchins for the first time was Oscar, Mr. Oscar Hertwig. His name was Mr. Oscar Hertwig in the year of 1875. He has described this particular process of fertilization in sea urchins. In the sea urchins. Okay, children. Now, coming on to, as I told you about the ejaculation, where does it happen? Okay, we have seen that this is going to be the ejaculation of the semen along with the sperms in the female reproductive tract, especially in the vaginal area, right? So it is going to happen there. But if you look at the fertilization process, so before I go into that procedure, let me first tell me that what all are being seen along with the sperm. So along with the sperm, we can see the presence of the semen, where you can see that this is going to comprise the 60 to 70% of the semen secreted by the seminal vesicles, right? So they are having the seminal vesicular semen, which is going to have the compositions like the fructose, okay, which are going to enable the sperm to produce ATP through their mitochondria. And we are going to even see the presence of the prostaglandins, which are going to bring about the contractions of the muscles in the uterus, as well as in the fallopian tubules, and you can see even in the vaginal area. And especially prostaglandins are going to bring these in the uterus as well as the fallopian tubules. And next we have another enzyme which is being called as coagulase, or we can even call it as vesiculases or vesiculases, because they are secreted by the seminal vesicular cells here. And children, you can even see the other one which is being called as a prostate gland. So the 30% of the semen is going to be from the prostate glands of the male reproductive system. And these are going to actually have the compositions like the fibrinolysin. They are going to have fibrinolysin enzymes, and they are going to have citrate, which are also going to fuel up the sperms. Okay, these all can be seen here. And just remember that these all, whichever are being present, like fructose. So what did I say about the fructose? Fructose is going to fuel up, right? So this is going to help in the production of ATP by your mitochondria, which are present in the middle piece of your sperm cells, right? And prostaglandins, especially, they are going to bring about what the contractions of the smooth muscles. They are going to bring about the contractions of the smooth muscles of your uterus, okay, uterus. And even you can see that even the fallopian tubules do perform the contractions, and especially uterine contractions are very powerful, and they are going to be especially the contractions of these uterine muscles, especially these are going to be backwards, okay, they are going to be backwards, and they are so strong that they are going to be either the sucking or squeezing effect they are going to give for sucking up these sperms here from the vagina. Okay, this is important. And children, if you look at the coagulase or the vesiculase, these are two are the same. Okay, and you can see that the vesiculase, what it does is you can see that the semen is going to be actually, semen is going to be a fluidy consistency, little bit viscous it is going to be, okay. But when it is going to be discharged into the female reproductive tract, so from the vagina, it may leak out. So in order to prevent the leakage, in order to prevent the leakage of the semen out of the vaginal area, what the coagulase does is, this is going to actually little bit, it is going to actually coagulate. These vesiculase enzymes are coagulating the uh, testicular fluid. Okay, the testicular fluid is nothing but the semen. Okay, the testicular fluid as well as a vaginal fluid, due to which, because of this clotting or clogging up, what happens is now the sperms are going to be latched. They are going to be attached to the what you call is a vaginal area. They are going to be latched out, okay, to the walls, lashed out to the walls. But if the sperms are not able to swim, then what is the need of this ejaculation? So then what happens is, here the one which is being called as fibrinolysin now comes into the role. So now what happens is, the fibrinolysin comes, and what it does is, it is going to release the sperms which are latched to the walls, right? Releases the sperms which are lashed to the walls of the vagina here, walls of the vagina near the cervix area. So now what happens is, the fibrinolysin, what it is doing, it is actually increasing the motility, right? It is going to increase the motility of the sperms, not to fix at one area, and now they're going to move directly into where, which surface, it is going to be towards the cervix area. This is going to be the one. So what have you seen is, the ejaculation is going to actually possess, what did I say? The 1 ml of semen is going to comprise of 10% of sperms, and all the other 90% is going to be the fluid of semen, as well as the enzymes, as well as the other components which are being present, right? Like citrates, the fructose, and all those things. So it is only 10% of the sperms over here. And you could even see that they were having fructoses, which are fueling up the mitochondria to produce the ATP, to help the sperm swim and make those movements. And the prostaglandins are bringing the contractions. The coagulase is coagulating to prevent the leakage of the semen. The fibrinolysin is bringing back the movement and releasing the locked out sperms in the coagulated uh, media. And the citrate is also going to fuel up the sperms. So these are all the things that you can see as the compositions of the semen which has been ejaculated in the vagina. Then speaking about your uh, ova. So how is this ova or egg, in which position or in which situation it is going to be present? Actually, the egg which is going to be ovulated, we all know that the Graafian follicle, the mature Graafian follicle is going to actually release the ova, right? And what is this procedure called as? This procedure is being called as ovulation. right? This is being called as a ovulation procedure. So if you look at this particular ovulation procedure, you can see that the ova which is being released here, okay, it is going to be actually the secondary oocyte. This is going to be the secondary oocyte, and this particular secondary oocyte is in a particular process which is being called as what it is under the process of metaphase 2. It's going through this particular phase, right? And that too, especially it's going to be meiosis 2, right? So when the ova has got actually ovulated, this ova is done, it has completed what? It has completed its first meiotic division. It has completed its first meiotic division, right? Now what is happening, what it is being called as, now it is being called as what? This itself is this, okay, this is a secondary oocyte. It's done with the first meiotic division. It is under meiosis 2 undergoing, and it is passing through the phase of metaphase 2, okay, metaphase 2, and this is still going on, it's going on over here. And now this is being called as what? Secondary oocyte or secondary ovum. We are going to call this as the secondary ovum, we are going to call. So your graphian, that is matured graphian follicle has released ova, which is called as ovulation, which is done with its meiotic 1 division. It is passing through the state of meiosis 2. Okay, it is passing through the stage of the meiosis 2, children. If at all the sperm, if the sperm is going to touch the ovum, if the sperm is going to touch the ovum, then what happens is the ovum is going to actually complete its meiosis too. So this is just passing through. But if the sperm touches it, what happens? If the sperm is going to touch, then it is going to result in what? The meiosis 2 division is going to be done or completed. Then your ovum will be called as what? It is going to be called as definitive ovum. We call this as definitive ovum. Did you understand this procedure? This is important for us now. So at the stage of secondary ovum or secondary oocyte, we can see that this is entering into where? Into the fallopian tubule. Okay, it is entering into the fallopian tubule, especially in the ampulla area for the arrival of the sperm. If the sperm comes and fuses with this particular what you call secondary ovum membrane, then instantly it is going to end up the meiosis 2 division, and then it will be termed as a definitive ovum. So this is the one which need to be understood about the ova. These are the prerequisites to be known before your fertilization, children. I hope this concept is clear for you all, children. So this as well as this procedure. Now what I'll do is I'll tell you the procedure, how they are going to be moving, right? So if you have seen, children, let me draw an ova which is going to be an almond shape, and you are seeing that this portion is bulged out because the graphian follicle is about to release the ovum. Okay, and you can see that the fimbriae are being present very near, so these fimbriae, and you can see that this is going to be your fallopian tubule, your ampulla area, and you can see the isthmus area, you can see the uterus. This is going to be your uterus, this is going to be your vagina, and this is going to be your cervical opening. So let me take this as a cervical opening. So let me tell you the procedure. This is very important to know that your ovum is motile. Is it correct? No, not at all. Ovum is immobile, or it is immotile, right? Then how does the ovum move from the ovary from here to that of the fallopian tubular area called as ampulla? How does it travel? Now this is becoming more interesting. So in order to teach these concepts, it becomes very interesting for me to introduce you with all these particular procedures. So if your ovum is immotile, then how does it travel? First thing, children, you should know that ovulation is under the influence of which hormone? Ovulation is under the influence of under the influence of a hormone which is being called as none other than luteinizing hormone, right? So there are some hormonal effects, influences on the female reproductive tract. So now what is happening is, because of the increased luteinizing hormone levels here, even they show the effect on the fimbriae. What are fimbriae? The finger-like projections. Okay, what are fimbriae? These finger-like projections. Let me color them and show you. So these are the finger-like projections that you can see. So these are the fimbriae of your fallopian tubule. So children, what you can see here is your fimbriae are going to become very active during they become very active during the ovulation process. During the ovulation, see the magic of the nature, that it is synchronizing few of the what you call is procedures or events that are happening at the same time during the production of the ova from the matured Graafian follicle out onto the surface. So let me draw this properly, then I can make it out. So this is going to be your ovary, right? So now the Graafian follicle has released the ova on the surface. So when it is going to release the ova, that releasing is being called as ovulation. At the same time of the ovulation, the fimbriae are becoming so active that now they start brushing, or they start sweeping. They start sweeping or they start brushing, like your mother does when you go to sleep, right? She brushes up your forehead while you go to sleep. So the fimbriae are also sweeping or brushing the ovaries during the time of ovulation under the influence of the hormones. Under what? Under the influence of the hormones. This happens only during the ovulation process. Okay. And secondly, this is first thing. Now secondly, what is happening is the fimbriae are going to have the fimbriae. Let me draw one fimbriae. So this is going to be the fimbriae, right? The fimbriae are going to be lined by the mucus cells. Okay, mucus cells, these are the mucus cells. Now if you look at the mucus cells, these are going to have the cilia. These are going to have cilia. So now what happens is the mucus cilia which are being present, they are also going to release some of the fluid, okay? Some of the fluid, and they're also, the cilia are also going to have ciliary movement, right? The ciliary movement, they're releasing some fluid. So you can see that the ciliary movements as well as the brushing movements and the fluid what you call is the fluid which has been released by these crunts, we can even call it as fluid crunts, the fluid crunts or the ciliary movements, the brushing or the sweeping of the fallopian tubule, that is fimbriae, will easily swipe off the ova into this particular procedure. Did you understand now how it entered into the fallopian tubule, especially into the infundibulum area, right? This area is being called as what? Infundibulum. Infundibulum area. So now it entered into the infundibulum area. Though it is immotile, it could enter into this. So now after this is going to enter into the fallopian tubular infundibulum area. Now the procedure of the things of events are going to happen here. Okay, so you know children that your ova is in a state of what? As a secondary oocyte with what? Zona pellucida. Very right, yeah. The So let me draw it over here also such that it will be easy for us. So we have a secondary oocyte cell with a haploid nuclei here. Okay, we are having the haploid nuclei here, but the arrested is, it is been arrested at what? It has been arrested at the meiosis 2, right? At the metaphase. At the metaphase, it is being arrested at the meiosis 2. Meiosis 2, it's been arrested. And this is having an covering which is being called as what? A zona pellucida. This is having what? The zona pellucida, which is a glycoprotein membrane. Okay. And out of these, they are having some follicular cells which are being called as what? The corona radiata. They are having these corona radiata cells also. So these are the follicular cells. These are the follicular cells which are being called as a zona pellucida. Okay, and these zona pellucida, that sorry, not zona pellucida, it's being called as a corona radiata. This is being called as what? It's being called as a corona radiata. So these are the corona radiata cells. And these corona radiata cells are being bound by whom? They are being bound by some of these connective tissues here. Okay, and these connective tissues here are going to have some chemical, and this is none other than here, this is being called as high hyaluronic acid. So this is being present here. So this is a high hyaluronic acid which is being present here, which is bounding up the connective tissue, which is binding up the corona radiata cells here. I hope you understood. This is going to be your secondary ovum that is waiting, where it is waiting in the ampulla area. And it stays for how many hours? It stays only for 24 hours in maximum women. Only in rare cases, the secondary ovum which has got released into the ampulla can even stay till 2 days, but at most it is going to be 24 hours only. So it only waits for the area. So time. So it has come to this particular area which is being called as a ampulla. Who is that? That is going to be your ovum. Ovum is going to be here. Okay, children, did you understand this one? Now, secondly, you can see that the ones here, the ovum which has been picked up here is being put in where? Which part did I say you? It's going to be picked up and put in this particular area, which is being called as here. Which one is this? The ampullar area. Okay, so that is going to be done. Now, if you are going to look at the next procedure, let me speak about the next procedure. Now, as we spoke about the ova here, the way it has traveled from the ovary to that of your, what we call is, it has come towards your infundibulum, then traveled into the ampulla. So this is also going to be moved by what, here it's going to be moved by the cilia, which is being present. Okay, let me write the other thing. From the fimbriae, now the third thing is from the fimbriae, the cilia of the fallopian tubule are helping in the movement, okay, as well as the peristaltic movements, of the fallopian tube, cilia as well as the peristaltic movements of the fallopian tubule togetherly are also going to move the ovum from your infundibulum to that of your ampulla area. Till here, did you understand, children? This is a movement of the ova. Now the ova has entered. It is sitting in the ampulla area. It is going to wait only till what? It is going to wait only till 24 hours only. It's only going to be 24 hours. So now if it is going to be the sperm, so remember that the ejaculation, let me take the black ink. So if you look at the ejaculation, where did it happen? It happened in the vagina, right? So the ejaculation has happened in the vagina. Okay, but what is a condition vagina? Vagina is highly acidic. For what reason? To kill the microbes, to save. Okay, but what is happening is just because of the high acidic nature, the motility of the sperm reduces. Motility of the sperms is going to reduce, and you have to note, children, that sperms will have a good motility or increased motility only in alkaline media, only in the alkaline media, which is seen where? Which is seen in the uterus as well as fallopian tubes. Uterus as well as a fallopian tubes. This point is important for us. I hope this is understood by you. So it's going to be acidic in nature. And secondly, you could even see that in the vagina, in the seminal fluid, along with the semen, what it come here, along with the semen, you could see that there was coagulase, or it was being called as vesiculase, right? So that is going to clot up the seminal and the testicular fluids. Did I write it over here? Let me show you. Yeah, I have written over here. So it's going to prevent the leakage, right? So now it is going to slowly get lashed out. They fix to the vaginal walls. Later, the fibrinolysin, which is a composition of the prostate glands, is going to release the sperms later. And now they enter into the cervical area. Okay, so they're moving slowly from here. So now you are understanding the movement of the sperms, I hope. So where are we? They have entered here, they have lashed out here. Okay, under the influence of what? Under the influence of whom? It was under the influence of the vesiculase. It got clotted and they lashed. Later, who released them? Someone released them. Who released them? It was fibrinolysin. Fibrinolysin has released them. So when it releases, what happens is, again, during the ovulation process, this is very, very important. During the ovulation process, the mucus secreted by the cervical area is going to be very watery, or it is going to have a very fluid consistency, which is going to enable the sperms to swim. So this is going to enable the to enter sperms to enter the uterus very easily, very, very easily. But during the other days, your cervical mucus is going to be very thick consistency, because of which the cervical mucus and the opening is going to be plugged up with this particular mucus. So it will not be easy for the sperms to make an entry into the uterus area here. This is very important. So in order to check whether a woman has ovulated or not, the test is going to be collecting the mucus from the cervical area and spreading it on what you call is a slide, and checking out the consistency, whether it is going to be fluidy, okay, and stringent. So if it's going to be very fluidy, that is going to actually give an indication that the woman has ovulated. Okay, so that is very important, children. So now they have crossed all this. They are using the fructoses. They are using ATPs. They are moving. Here the movement was very less, because this was very, very acidic. So the motility was very less, right, of the sperms. But later they moved here. Here it is completely alkaline. So the motility of the sperms is going to actually increase. Okay, so now they are moving towards the uterus. They are moving there. Now you can see that as it is going to move, the mitochondria are using fructoses, and they're having a very good tail propulsions. So now from the vagina, you can see that they entered into the cervix area, where you should remember that the cervix is going to have the fluid consistency mucus only during the ovulation. So now the sperms are being passed or being enabled. So now they are entering from the cervix into an area which is being called as uterus, which already I spoke that it is going to be alkaline in nature, which is increasing the motility of the sperms. Along with that, the fructoses are also helping in making the movement. So especially your sperms are using the tail propulsions properly in order to swim through the uterus. Okay, this is important. Another important thing that to be remembered during the uterus is going to be the prostaglandins. As I told you, that prostaglandins are going to enable the uterus contractions, right? This is going to bring the uterus contractions, and these uterus contractions are going to be like squeezing up, and it's going to be like sucking up action of the sperms. So it squeezes and sucks up the uterus now. And you can see that these sperms, they are going to move towards, okay, they are going to move towards the which area? The ampulla area. Okay, see, you have to remember that the uh, travel time, here I make a note, the travel time for a sperm for a sperm to reach your ampulla is going to be 5 minutes, only 5 minutes. Maximum it will take 45 minutes. Okay, this is very important to remember. And it is going to be only 1 in a million of sperms only can reach. 1 in a million of sperms only can reach. Only can reach 1 in a million. That's it. So it is totally how many? If it is 2 ml, it is 200. If it is 5 ml, it's going to be 200 to 500 sperms reaching. Where? To ampulla. To where? To the ampulla area. Okay, children. Now this is being seen. Secondly, you can see that even the cilia, right? Even the cilia of the mucus cells, even the cilia of the mucus cells also are going to help in what? The movement of the sperms. Movement of the sperms. Even they are going to help in the movement of the sperms. These all are helping in the movement, the tail movements, the tail propulsions, the increased motility of them, and the uterus contractions, and the cilia of the mucus cells, which are lining up the uterus. These all are helping it in to move. So now they will be slowly moving into which area from the uterus. Now they can easily go into, let me show you the figure. So from here it is being moved. Okay, now they come here and stop. Here they stop. It's a very interesting fact to know that after they move to the uterus, from the uterus, now they are going to go into the fallopian tube. And the first part of the fallopian tube is being called as what? Isthmus. They come to the isthmus and stop. Why did they stop there? Because they need a signal now. So once they're going to stop now, what happens is your ovum along with the corona radiator, very interesting fact here, that the ovum along with the corona radiator, what they do is they are going to throw a signal. Okay, they are going to throw a signal. What they do is they are releasing a chemoattractant. Same thing we have seen in the plants, right? When a pollen grain falls on the stigma. So the same thing is happening here. There's a chemoattractant. Okay. Now this chemoattractant is going to actually, it's going to make the sperm, signals the sperm to move ahead. Move ahead. From where, children? From the isthmus. So now they are moving. They are moving. Why? Because they have got a signal from the ova along with the corona radiator. They have released what? Chemical. What are those attractants? They are releasing the chemical attractants. Now slowly your sperms are swimming. They are swimming towards your ampulla area. So now they are moving towards the ampulla. Okay. But here I should speak about something else. The ampulla who is waiting, the ovum is waiting. But there is an interesting fact that before fertilization process, before the fertilization process, there should be two events that have to happen. The first event is being called as capacitation. The first event is being called as capacitation. The second event that has to happen is acrosome reaction. Acrosome reaction. These two have to happen. So now let us see what is capacitation. Capacitation. See, capacitation is nothing but before your sperm can make a movement towards the ova, your sperm has to have a head wash. Like you have hair wash. This head wash is nothing but in simple terms, I told you, but in scientifically you call it as it is conditioning of sperm. Conditioning of sperm in the female genital tract. This is being called as capacitation. Okay, that is capacitation. This takes 7 hours of time. Okay, where does it happen? Who does that? See, who does the capacitation? That is none other than the mucus lining of uterus. There we could see the fallopian tubules. Here also we can see the mucus, as well as the cilia of the mucus cells, as well as the enzymes present here. They all are going to wash up, okay, wash up or condition them. Okay, they are going to sperm condition the sperm. This is being called as capacitation. See children, for example, let me now take a sperm, okay? This is my sperm. I have my middle piece where I have all the mitochondria, and this is this, where you can see the acrosomal section also. And you can see that on the head of this particular uh, sperm is going to have glycoproteins. Now I'm taking some cholesterol, okay? Some cholesterol, and it is going to have some semen plasma proteins. Let me take another color, blue color I'll take, okay? So they're even having the semen plasma protein, semen plasma protein, okay? On the head, they are having some glycoproteins. Okay, they are having some cholesterol. Okay, next they are having some semen plasma proteins. Semen plasma proteins. These all are being seen. Okay, and they have this 23 number of chromosomes inside. Okay, so now what is happening is these all things will be washed away. By whom? By this particular mucus. See the sperm after the capacitation process. So here you have the mitochondria, here you're having the haploid nuclei. Now you can see that the glycoproteins are being washed away. The cholesterol is being washed away. The proteins are being washed away. Okay. Now this process is being called as the capacitation process, where the glycoproteins and all these things are washed away. Okay, that is being called as capacitation process. If there is no capacitation, remember children, if a sperm is not capacitized, what happens? It cannot fertilize. It cannot fertilize. Do you know how this was being actually discovered? During the in vitro fertilization process in a petri dish, when we take the ova, and when we release some of the freshly produced sperms, okay, there was no fertilization process. Later it was being discovered that what was that, why is the sperm not able to fertilize the ova, though it is freshly produced sperm? Why is it not to fertilize in a petri dish in the in vitro fertilization process? Later it is being discovered that the capacitation process is being carried out by the mucous linings of the uterus, fallopian tubules, the cilia, and the enzymes. These all are actually capacitizing your sperms, and later the sperm is able to fertilize. So after this discovery, now they have they synthesized some materials, and these materials, they efficiently capacitize the sperms, and now they are being used in the in vitro fertilization process. Okay, children, did you understand this procedure of the capacitization process? So this is an important procedure that you need to understand, that before the fertilization process, your sperms have to get capacitized. Okay, and this is happening where? This is happening while your sperm is moving in the uterus during the fallopian tubule. So the procedure is starting up when it is moving from the uterus only. Okay, and another important thing here I need to speak is about the males who are going to have a lesser amounts of selenium, who undergo the radioactivity, that is radiations, and who are going to be highly alcohol consumptions. These things, they are going to lead to what, they are going to lead to the double heads of the sperm. Double-headed sperms will be released during these procedures. Children, I hope in this video it is okay for you all till this capacitization process. So while it is moving, you could see the procedure that while it is moving, it is getting capacitized. Even here also this is getting capacitized. Right, children? Did you understand this? I have shown you the way the ova is moving into the ampulla area, the way the sperm is moving into this particular area. It is getting capacitized, removed off. See, remember, not all the sperms move, right? It is going to be only 200 to 300 sperms only making. What about all the other sperms? All the other sperms are going to be disintegrated, disintegrated and absorbed by the mucus of the female tract. Later they'll be feeded upon by neutrophils and monocytes. Neutrophils and monocytes are going to feed on them. So that is going to be the death of the sperms. Okay, feed. This is going to be the procedure. So children, to make this concept very clear for you, I have some of the slides. Let me show you those slides and end up the procedure today of this process till the capacitization process. Okay, so I'll come up with the slides now. Give me a moment for that. Yeah, so see, I spoke about that the mucus. This is a cervical opening. This is a cervical opening. Usually, before and after the ovulation, okay, that is not before and after the process. What happens is your cervix is almost being plugged up with the mucus like this. It is very, very thick, as you can see. But during the ovulatory phase, just before the onset of the ovulatory phase, what is happening is your mucus is becoming watery consistency to enable the movement of the sperms easily into the uterus such that they can move and swim through the uterus to the top of the ampulla. See, the periovulatory mucus is watery, facilitating the sperm movement through the open external os, which is nothing but the opening of the cervical or the cervix. Could you see this, children? This is the one. And next thing what I'll show you is a capacitization or capacitation, what we call it. So this is capacitation. As you can see that in epididymis, the sperm was this. Okay, later the seminal plasma. Along with that, you can see that the ejaculated thing is going to be something with the glycoproteins and all the other plasma, that is seminal plasma proteins present. They are this. But when they are moving into the female tract, because of the mucus, what has happened is all the glycoproteins and the plasma proteins as well as even the cholesterol, all these things are being cleared off, and this is being called as capacitated. So when the king goes to meet his queen, he is getting a head wash. Okay, this head wash is being done by the queen herself. Okay, that is going to be the female reproductive tract mucus is going to capacitate the male's glycoproteins and the cholesterol and the plasma proteins, which is being called as capacitation, which is very much important for the fertilization process to happen. Okay, children, let me see if you have, if I have any other sort of videos. Sorry, that is your, let me show you that. The other slides, if I have with me, yeah, of the ones. Yeah, children, let me show you this particular one. So it's a beautiful image where you can see the ova and where the hundreds of the sperms are approaching the ova, but it is going to be only the one which can make up the contact with the cortical area or the membrane of the ovum. Okay, children. I hope this has made sense for you all and understood this topic. If you like this video, please do hit like the button and please comment in the comment section and try to share and subscribe my channel. Do please comment how you liked it and how are the detailings of this video. Thank you all. Thank you.